Inositol Triphosphate as a Second Messenger by Samia and Eunbin Jennifer Cho.
Inositol triphosphate is an intracellular messenger that is formed after the activation of phospholipase C. There are three components to the inositol triphosphate system: the first messenger, the second messenger, and the cellular response. Inositol triphosphate is hard to pronounce, so some smart scientists shortened the name to IP3.
To further explain what second messengers are and their functions, we first have to look at the step before, first messengers. First messengers are extracellular ligands that relay or transmit cell signals. In this case, the first messenger is a hormone cell.
Inside of the lipid bilayer of the plasma membrane, there is a G protein coupled receptor. This signaling molecule, or hormone cell, binds itself to the receptor.
Along with the G protein coupled receptor, the plasma membrane also has a G protein, which activates mechanisms in the cell. The structural parts of the G protein consist of the alpha, beta, and gamma subunits, the alpha subunit being the active portion. The alpha subunit of the G protein connects to the opposite side of the receptor and dissociates into an alpha subunit and a beta-gamma complex when the GTP binds itself to the alpha.
Associated alpha and GTP then fall off and interact with the membrane-bound enzyme, known as the phospholipase C, by binding to the left of it, which activates the enzyme.
Once activated, phospholipase C then acts on phosphatidylinositol 4,5-biphosphate, or much easier, PIP2, and breaks down the lipid into two components. In a sense, phospholipase C cleaves PIP2 in half. The top half becomes diacylglycerol, or DAG, and the bottom half becomes IP3. The DAG stays attached to the plasma membrane.
IP3, the second messenger, then diffuses to the calcium channel that is embedded in the smooth endoplasmic reticulum membrane. In general, when the first messenger aspect is activated, it initiates a pathway that leads to the activation of the second messenger response, where the second messenger is released.
Once the IP3 binds to the calcium channel, it causes it to change shape and open up. The channel splits apart, creating an opening that allows the calcium ions to pass through the smooth ER into the cytosol.
When the concentration of the calcium ions become greater in the cytosol than the smooth endoplasmic reticulum, then the IP3 will disconnect from the channel, therefore closing up the calcium channel.
The channel allows calcium to travel down its concentration gradient as they flow outside of the endoplasmic reticulum, increasing cytosolic calcium concentration levels. These calcium ions activate various proteins, which ultimately leads to a cellular response.
An example of one of these proteins is protein kinase C, which is a protein that calcium and DAG activate. Once this protein is activated, phosphorylation, or the addition of a phosphate group, for target cells occurs within the cell, causing a cellular response.
IP3's role as a second messenger in this system is vital to the survival of eukaryotic cells. This process and IP3's part in opening the IP3 or calcium channel is critical to the survival, propagation, and function of cells because calcium has a role in allosteric regulation on enzymes and proteins once it enters the cytoplasm, which allows cells to function normally.
Thank you, and we hope we taught you a little bit about the amazing things that cells can do.